But here we get to talk about base quality score recalibration. And I'm reminded that I have to set my timer because I like to ramble. Okay, let me do that right now. Okay, set. Okay, so we're going to talk about base quality score recalibration. And quality scores are like my favorite thing in the world, so this will be a lot of fun. So the thing is, I told you that this machine is going to spit out these letters, right? A, C, T, G, whatever. But right alongside it, it's going to spit out a quality score, which is the estimate of the basis confidence, right? And these are important for a lot of reasons. Oh, I should say that we are, as you might have guessed, we're at the next session here. We're going down this pipeline, we're zooming right along, and now we've arrived at base quality score recalibration, and we have one more session this morning, and then we're done. Okay, so the thing is, I told you that calling variance is hard, right? So you have this data, it's quite messy, and what do we think about this thing in the middle here? It's, there's some evidence, some green bases here, which you know from IGV those are the bases that don't match the reference. So maybe there's evidence here for a genetic variant in this sample, right? But what if I told you that those bases that have that alternate allele, what if those had low quality? What if every time the machine read a green base, it was actually only 50% confident? You'd be pretty unlikely to say that's a real variant, right? So these quality scores are like the foundation for allowing you to do proper statistical estimation of the evidence for variation, right? And so if these quality scores are wrong or biased in some way, you're gonna falsely infer the presence of genetic mutation, right? And so these quality scores are incredibly foundationally important. And so we have this huge procedure called the base quality score recalibration, which tries to make those scores as accurate as possible. Because what happens is those quality scores, as they're reported from the machine, are actually quite bad. They're quite biased in many fun, crazy ways. And so this is my introductory example to show you how it can be biased. And so what we have here are plots by dinucleotide context. So you read an A followed by an A, or you read a T followed by a C. It's a two-base context, and that's what's plotted on this axis. And on the other axis is the accuracy of the quality score estimate. Right? So what this plot is showing you is that every time the machine read an A followed by an A, it overestimated its quality score. But if it happened to read an A followed by a C, it underestimated the quality score. And that kind of bias is just crazy if you're trying to infer genetic mutation, right? So you'd be way more accurate to call it as a variant if it was AA, but less likely if it was AC, right?
What are you comparing it to that you know it's over?
Yeah, so we're gonna get there. We're gonna. This is sort of like an introductory thing to let me excite you that these are really biased. So I'm going to explain how we estimate the accuracy, and you'll see that as you go along. But take it on faith for now that we know the empirical quality of the bases, and you know the reported quality because that's what was just written in the file, right? And so you can do a delta and show that you're either overconfident or underconfident. But the thing that's important is that it's grossly biased by sequencing context. And so you're sort of grossly biasing your mutation calls by a silly thing like context, which is maybe that's biologically relevant. And so well, anyway, I think you get the picture. So we have this procedure called the base quality score recalibration. You run it, and what you get is this plot over here, and this bias has been beautifully removed. And so you can estimate that using the root mean squared error. So we have a very high number over here. We run the base recalibrator, and now we have a much lower number. And so as a result of using this procedure, you've sort of removed this bias in the quality score estimates. Pretty good? Okay, what's up? Oh, it absolutely is. So I have lots of plots of lots of different kinds of biases, and it's absolutely a problem with the sequencing device, right? So they've spit out a string of quality scores, and those quality scores are not accurate. So perhaps we could convince them to make them more accurate. I don't know, or whatever. The thing is, maybe these are for-profit companies, and they're incentivized to say their quality scores are better than they are. I don't know. Who knows, right? they can spit out whatever they want. Maybe they like spitting out the number 30, and maybe that's biased in some ways. Like they can do whatever they want, but we can fix it. That's the thing. Make sense? Okay. So how does this work? Well, so what we're going to do is, I told you about the physical device and how it's separated by lane. So what we're going to do is we're going to count up all the data as a function of several things, as a function of the lane, as a function of the reported quality score, of the position within the read, of these sequencing contexts like I showed you, and we're just going to count up the data, and we're going to say every time we saw Q30 followed by AA in this lane, we saw it 1,000,000 times, and it was an error 10 times. So that 10 over 1,000,000 turns into a quality score, and that's the empirical quality of that set of data. So all this procedure is just simply a piecewise linear tabular thing where you're just counting up how many times you saw that combination of data, and because you have a whole sequencing, a whole lane's worth of data, you have millions of observations of that particular combination of covariates, and you can infer what the actual quality of that data is. Okay? And so here's how it really works. We're going to, like I said, we're going to stratify by lane, quality score, machine cycle, and sequencing context. We're going to use, luckily, thanks to having dbSNP and 1,000 Genomes Project and all these things, the vast majority of the variation in your sample is known. So you simply discount those sites, you kick them out, and you say any other like mismatches in the read, those are the errors. And although that's it's kind of a little bit of an overestimate of the error, what you're doing is you're trying to find these like gross biases, right? And some really powerful things happen when you do this. So you take, just like I said, you take the number of mismatches divided by the number of observed bases, and we have a little fancy smoothing thing just to help us with bins that have small numbers of counts, and we turn that into a quality score. And that's going to be the empirical estimated quality score for that combination of data. Make sense?
But I mean, so mutation rates can have bias based on, say, time and time.
Yeah.
How do you account for that if GC is likely to say more likely to actually be mutation?
Well, so what we're going to see is that we're trying to find like, oh, well, actually, why don't you say it one more time? Make sure I repeat it correctly.
It's for the recording.
Yeah. No, but I want to.
So if the mutation rate at certain positions can be based on some of these factors, such as sequence context, then how do you know that you're not actually biasing your calls by just assuming that everything that's mutation is de novo is an error?
Yeah. So the question is, there's actual like real biases in the variation. So how do we know that we're not like discounting that? So the point here is that we're trying to find like really gross biases. Like it'll be obvious that like when you have cycle 5, there's always a mutation, right? Like it's like seriously not biological. It's like so obvious it's not biological that it just jumps out. You know what I mean? And those are the sort of gross biases that we're trying to fix with this sort of thing. Like having a minor fluctuation in GC versus whatever, that's not what we're trying to fix here. What's up?
So if you're working with non-human data, you don't have something that's like, well, annotated in the script, so if you're going to say like active Excel spreadsheet or area variation, is there any way that you can sort of like make the file?
Yeah, of course. So the question is, what do you do if you don't have dbSNP or a database of known variation, or you're working in a sample that's not human, so you don't have all this beautiful data? So the answer is yes, there's tons of things you can do. The whole idea is you just want to sort of discount the sites that are likely variant, and the things that are sort of random scattered errors, you want to count those as errors. So there's tons of things you can do. You can take your Excel spreadsheet, you can convert it into a BED file, and just feed that in. You can convert it to a VCF file, feed that in. You could do a first pass calling of variation, just a really like simple, like give me the list of variants based on the data as it is, and feed that in as your database. You can do tons of things. The whole point is you just sort of want to discount the things that are likely variation And the things that are just sort of scattered random errors, those are the things we're going to count as errors here. And in aggregate, that information, when it's aggregated together, becomes really powerful. Even though it isn't perfectly accurate, because some of those things really are variation and aren't error. But it's just that combining together of the data to find the gross errors is really powerful. Does that make sense? Hopefully I'm...
In some process, you don't lose the data, you're just changing the quality score.
Right. So what's going to happen is the BAM file is going to be exactly the same. The only difference is the quality score field is going to change. Yep. So instead of having Q30 somewhere, you're going to have a Q40. Instead of having a Q45, it'll change to a Q33, and that's the only difference. Yep. And in fact, if you want to, you can keep the original quality, and you can use both of them somehow if you want to be fancy or do whatever you want. Okay, so let's see what it really looks like. So this is my favorite slide because it shows that, first of all, there's a lot of data, but it also shows that I'm not biased by any particular sequencing company because they're all pretty bad. So, so what's going to happen is I have dots. I have purple dots and blue dots. So the purple dots are the original data that came off the machine, the reported quality scores as they came off the machine, and the blue dots are after you've run it through this base quality score recalibration procedure. And what I'm plotting is on one axis will be, for the top plots, we have the reported quality score, and then in like a particular bin, if you take all the things that are reported at Q20, you can ask what is the aggregate quality score of just those bins. And so what you see is that when you're Q20, in fact, it's really Q17 or whatever. So maybe we'll say that this company overestimates their quality scores. They say they're overconfident in how good the data actually is. So when it's Q35, it's really Q29, and that's a pretty big difference actually because the quality scale is a logarithmic scale. So the difference between Q35 and Q29, when you're sort of like you have like 4 reads on your sample or 10 reads on your sample, that's actually quite a big difference in your confidence in the variant. Now, so some really fun and exciting things come out of this. So you could maybe think that other companies are doing a better job or maybe they're doing a better job of estimating their quality scores. Maybe they've tried to make it match the original, but by whatever they did to do that, they've added biases in other ways. So instead, if you plot the data by machine cycle, and so machine cycle is just, it's as you read along. The machine read an A, read a T, read a C, and that's cycle 1, 2, 3. You just read it along. And it has crazy, crazy biases as a function of the machine cycle, right? Like in the beginning of the read, you're like overconfident, and then you're fine, and then you're underconfident again. It's just crazy, right? And so if your variant like appeared at the beginning of the read, if all the reads that support your variant, if it appeared at the beginning of the read, maybe you'd be less likely or more likely to call it based on this bias, right? And so you can see after running this base quality score recalibration procedure, that bias has been effectively eliminated. Is everyone convinced? Okay, and I've already showed you by sequencing context, and it's just wild. Okay, so what's been exciting over the past maybe year has been that what we've discovered is that this procedure, not only does it work for SNP mutation, but the exact same logic and procedure can be run with insertion and deletions. And you can actually estimate the confidence, you can estimate a quality score for an insertion quality score and a deletion quality score. So what I'm doing here in this data is I'm using that exact same procedure where I'm counting up the number of times we saw an indel and how many times it mismatched, how many times it wasn't in the dbSNP or in your 1,000 genomes database or any of these things. And you can estimate the error rate as a function of, here I'm plotting an 8-base context, right? So you read 5 A's followed by this suffix. So it's an 8-base context, and I have 2 sets of data here. I have HiSeq data and Pacific Biosciences. And what you see is a crazy difference in the bias, in the insertion and deletion bias as a function of the sequencing context, right? So this HiSeq machine, the indel quality scores are much, much higher because it doesn't make many indel mistakes, right? It's Q40, so it doesn't make very many insertion and deletion mistakes, but those mistakes have a crazy bias as a function of the of the sequencing context, right? So if you're 8 A's or 7 A's followed by a C versus 7 A's followed by a G or whatever, oops, there's crazy bias. But to contrast that is with the Pacific Biosciences, where it makes a lot of indel errors. Each indel actually only has Q10 accuracy, which is quite bad, but it's beautifully unbiased by sequencing context. Does everyone see the difference there? So then the other thing to point out is that I'm showing you in the HiSeq data, there's 8 dots. They're differently shaded in green. And what that's showing you is that the per lane variability. And so that's why it's so important to create an error model that's specific to every lane. Because you can see that the darker lanes, they have less error in this context, but the lighter lanes have more error in that context. So it does vary quite wildly from run to run of the machine. And so capturing that is important. Okay? Why only 3 what?
Extra letters.
Oh, it's just to make it viewable.
No, but I'm saying maybe you will have a different mistakes with different contents if you look on a longer
Yeah.
suffix.
Yeah. So what happens is when we run this, you're actually using the full 8 base context. So every combination of 8 letters is a different bin, and you're calculating the data independently.
Yeah, but why not 10 bases bin?
Well, it's just a computational trade-off. So eventually you spread your data out so much that each bin has not enough data to estimate the confidence, and so you have to stop somewhere. So that's just the way we do it. So I think for indels, the maximum we would go up to for like a run of HiSeq would be 8 bases. Because 4 to the 8 is pretty big, and so you're spreading your data out a lot. And so So potentially there's some structure in the data at 10 bases, but you spread the data out so much that maybe you can't see it anymore. That sort of thing. Does that make sense? The trade-off? Good question. Okay, so here we're showing, it's just another example showing that we have, so the base quality score recalibration procedure, it's estimating not only the base substitution error rate, but also the base insertion and base deletion. And again, you can see these biases by cycle that are just wild, and they're removed by this procedure. Okay, so now let's talk about how you actually run it. We're going to do command lines and stuff like that now. And so there's 3 pieces to this thing. There's you're going to do base recalibrator is how you build the actual error model. You calculate the data and you actually build the model. And then when you want to apply the model, you run this thing called print reads. It's very simple. You just print the reads back out, but with the recalibration file to tell you how to change the quality scores. Right? So there's one step to build the model, and then another step to apply the model. And then we have this third thing. If you want to view if you want to make the sort of plots that I've put into this presentation, you want to view the before and after picture and make sure things are looking good and how it looks, like make sure that the bias has been removed afterwards or see what it looks like. Then we have this other procedure called analyze covariates just to make these plots for you, and it's just a plotting tool. Okay, so let's go through them. So there's going to be 2 procedures. So one is if you want to just process the data, so you run base recalibrator and then print reads and you have data. But if you want the beautiful plots, then you have to do before base recalibrator and after base recalibrator. Then you feed those 2 files into analyze covariates and then you get the beautiful plots, right? So you need the before model and the after model to build the plots. That's how it works. Thought I saw a question, but maybe not. Okay. So here we have the workflow. So you take in the BAM file and your database of known variation, and you run this thing called base recalibrator, and what it's going to print out is this recalibration table, and that's the actual error model. And so it looks like this. We've seen the command lines before, so we call it base recalibrator. We still need the reference. You have the input data. The thing that's here is the known sites. That's how you're going to give it the VCF files or the bed files or whatever you want to have the database of known variation. And the output is a table. And then now you have the table, and you're going to apply it with print reads. So you give it the original BAM file, and now the table, and you're going to get a recalibrated BAM file. All the reads are the same, but the quality scores have been changed. So that's called print reads. Same thing, reference, input, and you have this BQSR, which is the input table, and now you're going to write out a thing. Make sense? The only thing to say here is that if you want the original qualities, they're going to be there in a special OQ tag. In case you don't trust me and you want to go back to the original qualities or whatever you want to do. That's that. So now let's talk about how to get the plots. And so it looks like this. So it's basically two passes. You have to do it once to get the before model, and then you have to follow all these squiggly lines, and you do it again to get the after model, right? So you have the before model, the after model, and you feed those both into analyze covariates to get the plots. So we've seen this before. That was what I showed before, and then you do it again, and that looks like this. So base recalibrator again. You have the reference, input, the known sites, and now this time here you're going to pass in the before model, the BQSR. That's the before model, and then what you get out is the after recalibrated model. So now you have two models, the before and after, and you feed those into analyze covariates. And so before, after, and then you get the plots. So these are just going to be, it uses R to build PDFs. You have to have the right version of R installed and all those special things, but that's pretty simple. And you get the PDFs, which are the plots that I've put into this presentation. That's really it. Let's see how we're doing on time. Beautiful. And how do we know if it worked? Well, how you know if it worked is that you just look at these plots. We've wanted to, the best way to show someone how this thing works is you just show the bias, right? You show this crazy bias and you show that it's been removed. And so that's why we have this whole extra rigmarole to get these plots, and everyone should be looking at them as a matter of your QC, standard QC process should be, part of it should be looking at these plots and making sure the data looks fine. And that's it. Any questions about this procedure?
Yeah, this recalibration done at the FASTQ level, so it sounds like it's an independent process from the line.
Yeah, so unfortunately it's not, because what happens is you're going to take the data and you're going to say, this SNP at location 10 million is in db SNP. And so all of my reads that have a SNP there, you don't want to count that as error because it's real variation. Whereas if you're aligned to a place where there's one isolated mismatch and there's nothing else around it, you want to count that as error. So it does actually need the aligned data in order to make
themselves, just looking at mismatches, you don't know if they're mismatching being aligned.
Yeah, exactly. So you need the pile of evidence to tell you that it's mismatching and.
And local realignment seems to be important for this recalibration to work properly.
Exactly. So what you saw is that you saw a slide from Ballantine that had crazy SNPs, right, all lining up. And so maybe you would think that's real variation, or maybe you'd count that as error. But when you have the correct view of the data, which is an indel, you would not count that. That wouldn't show up in your tables as error, and so that would affect the bias. What's up?
Any data or sense of just how many, when you get down to the variant stage, how many differences it makes, how many changes?
It's crazy, man. It depends on your project for sure. If you have a billion X coverage of your exome or whatever, maybe it's not so important. Who knows? But if you definitely if you have 4X coverage of your read, you're trying to weigh that's some seriously hard statistical analysis, right? And so properly having those quality scores, the difference between 99.9% confident or 99.95 is pretty huge in that sort of case. But the other thing to say is that if you do have these catastrophic biases that I've been mentioning, like someone turned off the machine and turned it back on and they misincorporated a base at a cycle, the only way to get rid of that sort of thing is to do this sort of procedure, right? And see that cycle 5 has an error every single time. So the quality score of every base in cycle 5 better be Q0 because you have no confidence, right? And so So if you're worried about catastrophic failures like that, you have to use this no matter what. There's no other way to get around it, really. So anyway, it's huge. And if you want to call indels at all, you definitely need this error model to do anything, because indels are hard. Okay, what's up?
For the plots, you have like a recalibration for insertion and deletion. As I know, you don't have quality calculation because you only know that control this here you have deletion.
Yeah.
And how you can find that plot?
It's fun. So.
How
So the question is about deletion qualities. So how can you know, you don't have an actual base there, it's been deleted, right? So how can you like assign a quality score or something? So it's been fun. So we've invented this thing called the base deletion quality score. And it's like my personal crusade to make this like a real thing and convince Illumina or something to estimate their deletion quality and output that, right? So what happens is the quality score, the way it's encoded is, it's what's the probability that the next base in the read was misdeleted due to slippage or whatever reason. So it's like it is a per base quality score based on what's the probability that the next base was accidentally deleted, right? So it doesn't matter that it's not aligned to the genome or whatever. It's a per base estimate. It's like you're asking the machine, what's the probability that you accidentally like did a slippage event or you did a whatever misincorporation, right? So it's really a physical property of the sequencing device, not a property of the genome or the reference or anything like that. So that's why you're able to estimate it on a per base level. Like you're asking the machine, did you think you made a mistake or not?
So which means the input base quality is not about plus Q file instead of you make it that and then you recalculate.
Exactly. You're right. So the question was, for the base qualities, you have these reported qualities from the machine. What's the probability that you had a mismatch or whatever? But for these insertions and deletions, you don't have that evidence. You don't have the machine's actual evidence on its own until we convince them to add it, right? That's what's so beautiful about this. Once we convince like PacBio or whatever, because PacBio internally, the machine does know it's evidence that it made an insertion or deletion mistake. And they just don't output it because it's not part of the file format. But internally, that machine is estimating its confidence that it did a slippage event. And so eventually we can incorporate that. But right now, we just sort of have like a flat prior. And so we base it, so for indels, it's more heavily based on cycle and on sequencing context and not on the reported quality score in the machine. Whereas for substitutions, it's more based on the estimated quality coming from the machine. It's really interesting stuff. What's up? Yeah, so the question is about coverage and how much coverage do you need? And really the answer is that the more is the better, right? Because the more data you have, the more accurately you can estimate these bins, right? But the thing is you have tons of control. Like you can decide that you don't want to cut up the data so finely if you don't have so much data, right? If you only have 4x coverage of a single lane, maybe you can't do a full 8 base context because you don't want to divide your data that much. So instead, maybe you'll do a 3 base context or something like that. So the answer is you basically need a lane's worth of data, right? So maybe that's I don't know how many gigabases. It's probably like 1x coverage of the genome or something, right? It's like a lane, and so you do need a lot. There's no question about that. You need a lot of data, but it's like I don't know. It's like maybe 1 gigabase of sequence data. It doesn't. So the question was, what about read length? So this is my alarm telling me I'm over time. So the question is, what about read length? And so the reason why read length doesn't matter is because you're not, the read length is not like a covariation of the error here. It's like you're sort of treating each observation independently and you're just throwing them into a table. So. So the question is, So bigger reads would have less data, yeah, because so you're estimating not only the error in like the machine, but you're also estimating the error of like the aligning process and all the processes that came before it. And so yeah, you would see that longer reads maybe they're able to align better, so they have fewer errors. So yeah, that's definitely true.
I bet the G2s with 36 bases and the I6 with 76 on their slide.
Potentially, yeah. Yeah. Yeah, longer reads still have errors though. I mean, and they still have like catastrophic biases as well. Before we do more questions, I'm going to ask Geraldine if we're okay to do more.
If Eric can be fast, yeah. Eric talks fast, so.
Okay, so this is fun stuff. Let's do more questions. What's up?
So I did an experiment where I had like 48 samples pulled in one lane, and now they're individual NAM files.
Yeah.
For me to put them all in one read group and combine them all somehow.
Yeah, so that's interesting. So yeah, so what he's asking about is like this sprinkle barcoding approach, if people know that term. But basically you have a lot of samples mixed in a lane, so you're sort of divided up your data a lot, right? So a lane is like a fixed sequencing budget, and you've divided that amongst a bunch of samples. Okay, so my answer would be yes. The best thing to do is to pull that back together and do this process per lane, because then you have, although that's complicated, you have all the data that you need to like infer the error, because the error is like isolated to the lane, right? Like so yes, the best thing to do is to do that. But if you have enough data without doing that, then that's fine. But if you don't have enough data, then the right thing to do is to pull that back together and then do this procedure. It doesn't need to be one BAM file. It just needs to be tagged with the same read group. It's not easy, but there's ways to make it happen, I'm sure.
Yeah, is the best practice workflow also working for amplicon sequencing? So far you're talking about whole genome, WGS or WES. For amplicon sequencing, targeted sequencing, do you have a separate best practice workflow?
I see. So the question is, what about capture sequencing? And so the answer is that the best practice is basically the same. It's just a matter of whether you have all the things that you need, like whether you have enough samples and enough variation to do the variant recalibration procedure. You have to decide that on your own. Whether you have enough data to do base quality score recalibration, it depends on, if you're doing the whole exome, then yeah, that's a whole lane's worth of exome data is enough. But if you have like, one gene that you're targeting, maybe you don't have enough data.
For example, many cancer panels only target 46 genes.
Yeah.
Rather than the whole exome.
Yeah, so maybe that's not enough data. Maybe that's too little. I mean, and the best thing about this is all you have to do is try it and look at the plots, and you'll be able to tell for yourself if it's working.
I think I'm talking about amplicon data is quite important.
Yeah, exactly. The PCR.
Yeah. Okay. I think there might be a section tomorrow about like specifics for like non-human data and specifics for like targeted sequencing data. Okay,
Next
no.
time.
Okay. Well, maybe we'll have a little working group where all the non-human people can get together and Okay, we're good to move on?
Yeah.
Thank you so much for your attention.